How is it that we find ourselves surrounded by such complexity, such elegance?
The genes of you and me, the genes of you and me, they're all made of DNA. We're all made of the same chemical DNA. We're all made of DNA.
Hi, you're listening to DNA Today, a podcast and radio show where we discover new advances in the world of genetics. From genetic technology like CRISPR to rare diseases to new research, we have you covered. For a decade, DNA Today has brought you the voices of leaders in genetics. I'm Dineen. I'm a certified genetic counselor and your host. I was recently a guest on the Science of Everything podcast. The host James and I had a fantastic discussion about genetic testing. This might be my favorite episode of a podcast I've ever been a guest on, which is why James and I decided to also release the episode on the DNA Today podcast feed. So to give you a teaser, we covered a range of genetic testing topics from the process and science behind the testing, types of conditions that can be tested for, and the difference between genotyping and sequencing. We even discussed direct consumer genetic testing like 23andMe and Ancestry, including the potential and current uses for criminal justice. We speculated about social implications of the testing, along with other futures that may be in store for genetic testing. So hope you enjoy this episode. If you do, and you like James' interview style and his cool accent, be sure to subscribe to the Science of Everything podcast.
Welcome to the Science of Everything podcast. Today we're doing a special episode. My guest today is Kira from DNA Today. And Kira, welcome to the podcast.
Well, thank you so much, James, for having me on.
So Kira, tell us a bit about yourself and your podcast and sort of what you do over there.
Sure. So my name is Dineen, and I am a genetic counselor. So we'll get into kind of what a genetic counselor does. But in my role, I meet with pregnant patients or patients that are looking to become pregnant in the near future to talk about genetic testing and family history. So all kind of genetic conversations there. And then on DNA Today, I'm the host and producer of the show. I've been doing that for 10 years. And it's been a blast. I talk to a lot of experts in genetics, talk to them about whatever they're researching or whatever they're an expert in. So it's a really, really fun show. And we've had a lot of episodes over the last 10 years. So it's a great way to just be active in the genetics community outside of direct patient care.
Yeah, that's really cool. It's exciting to meet a fellow podcaster who's been going for so long. What motivated you originally to start the podcast, and what sort of kept you going for so long?
In high school, I learned about genetics, started to, and I was like, well, this seems like a cool area of biology. And I was like, I want to do something in genetics, but I don't know exactly what a career in genetics looks like. So I started by starting the podcast as a way to like explore different careers and kind of get my feet wet with genetics, and learned about genetic counseling through that. And then was like, all right, I want to become a genetic counselor. And so the show really, in some ways, is kind of like, for me, advancing through my career of high school to college to graduate school, and now being, actually full-time employee in genetic counseling and everything. So it's a lot of interviews. When I started out, I truly did not know anything that the guest was going to say. It was like I was interviewing them for myself, and there were some people that would listen. And now sometimes I have an idea of what the guest might say, depending on what we're talking about. But yeah, it's just been really fun to be able to just network with so many people in the field. So I think that's what's kept me around and still producing episodes, but also just having a more engaging and larger audience as time goes on, and becoming a business for myself too. So I think there's just a lot of ways that I just love podcasting. And so I'm certainly kind of committed to the field at this point.
Yeah, that's awesome. So today we're going to be talking about genetic testing, sort of broadly, and might branch out a little bit from there. This is sort of related to the area that you work in, I guess. So tell us a bit about what is genetic testing.
Yeah, so there's so many different types of genetic testing. In general, when we're looking at genetic testing, we want to see, is there any genetic changes in a person that could increase their risk for a condition, or that diagnoses them with a condition? Or if there's a possibility that future children, biological children of theirs, could have a condition. So really being able to look at risk levels or give it a diagnosis. So with genetic testing, we're looking directly at our DNA, so the code of life. And so we're looking at this code and saying, okay, is there anything that's different there? And if it's different, Does that mean that it's different and it makes something in the body not work? Or is it just the beautiful aspect of human diversity where everybody's a little bit different? So genetic testing is very interesting, and it's come a long way in the last few years. So I think that's an interesting part, just to look at it from a historical context of just how expensive it used to be and how inexpensive it is now.
Yeah, it's pretty crazy how much that technology has advanced. And I want to talk a little bit more about that later. But I want to start with delving into some of the science a little bit more. So we've talked about genetics a few times on the Science of Everything podcast. I think it's been a while since I think it's been a little while since I did one. And we haven't talked about genetic testing though, which is one of the reasons I thought this would be an interesting topic. In addition to, obviously, that's what your background is. So one thing that we know is that DNA consists of a series of nucleic acids, and we know that DNA codes for proteins, which are molecules that carry out many of the key functions in our body, and that differences in genes between different people can code for, well, sometimes, as you mentioned, just sort of fairly trivial or unimportant differences, or sometimes they can code for disease traits or other problem areas that could be an issue for us. So one of the things that I wanted to ask about is sort of how genetic testing works. So it's obviously they take, I assume it's a blood sample that they take, and I'm curious as to what sort of tests exactly. So are they doing a full genetic sequence, or are they just looking for particular markers? Like, tell us a bit about how that works.
Yeah, so it definitely depends on what the healthcare provider is ordering. And you said, blood sample is usually a common way to collect a sample in order to actually perform the genetic testing. But a lot of testing can actually be done through saliva. So some people might be familiar, with like you get a kit and you spit into it and send it off to a laboratory. So there's testing that you could just do through saliva, because your cells throughout your body, except for a couple exceptions, all have the same DNA in it. So I can have blood from a patient or saliva, and I can do a lot of the same testing. Sometimes you need blood for a certain reason, but in general, for a lot of genetic testing, you can have either. And usually saliva is a little bit easier to get. You don't need a phlebotomist for that. But your other question in terms of, are we looking at entire genes? Are we looking at hotspots? It also, depends on what the provider is ordering. So, sometimes the largest genetic test is going to be the whole genome sequencing. So this is a test that is actually reading through all of your genes in your body. So this is, you imagine how much data that is. That's a lot of data to sort through. But that would be like the biggest.
It's about 3 billion nucleotides. Is that right? If I recall correctly.
Yeah. So that's a lot of letters to read through, right? And the next, the tier below that would be whole exome sequencing. So this is just looking at a small percentage, about 1% of your DNA that is actually active. It's being actively transcribed into mRNA and then translated into protein. So these are the genes that are actually made into proteins and actually doing something in your body directly. So a lot of testing now is whole exome sequencing, because we're like, well, let's just look at the active genes. Is there any difference in those that we need to be aware of?
Okay, yeah, this is really great. So a few more questions here. One that I have, okay, so two about the source, one about blood and one about saliva. So one thing that I've kind of wondered, and I guess I could have looked this up, I just sort of haven't got around to it. When they take a blood sample and if they do some genetic tests on that, or using that for, say, forensic genetics or whatever, where do they actually get the DNA from? Because erythrocytes, red blood cells, don't have DNA. So obviously they're not getting them from that. Is it the white blood cells? Like, where does it actually come from?
Yeah, you got it. So as I said, there's some exceptions that some of our cells don't have all of our DNA. And as you brought up, red blood cells don't. They're a different type of cell, I guess. So what we're able to do in the laboratory, and I've done this, and it's cool that you're able to basically separate out that blood sample into red blood cells and white blood cells. So in the white blood cells, you're able to actually get DNA and process that. So that's the actual cells that are being analyzed, for a lot of the testing, especially in the lab. I used to do what's called a karyotype. So that's a different type of genetic test where we're actually looking at chromosomes. So our DNA is like the bottom level. So going up from that, chromosomes are made out of DNA, and we have 46 chromosomes, 23 from our dad or biological father, and 23 from our biological mother. So when I would do karyotypes and looking at someone's chromosomes, seeing if there was anything different there, I would break apart and say, all right, I just want the white blood cells, that buffy coat layer when you kind of have the different layers in the tube. And that's what we would use to actually look at the cells and look at the chromosomes.
Yeah. So the layers come from centrifugation. Is that right?
Exactly. Yeah. So you
that's
spin it down.
Oh, sorry, yeah.
Yeah. So you just, obviously there's a few steps to it. So, but you're spinning it down so that you're using gravity to separate the different blood cells.
Yeah, I have done. I don't think I've worked with any human tissue before, but I have done a little bit of that in the lab. I think it's interesting to sort of think through how this works in practice. Okay, so that's the blood tissue. Now about saliva samples. So what actual cells are in our saliva? Like, I actually have no idea there. I assume saliva is mostly water, which obviously that's not going to have genetic material in it. So yeah, so where does the DNA come from there?
So I think it's mostly just from like cheek cells.
Yeah.
So sometimes, like I know I've done like a parentage testing where we're seeing, okay, is this a biological father or not? In those tests, we take a cheek swab. So we're actually taking like basically like a really long Q-tip, and then going on the inside of their cheek and then swabbing each side for, I don't know, whatever it was, like 10, 60 seconds or something. For each side. So that we're like trying to directly get cheek cells.
Yeah.
But one of the things with the saliva samples is you can't eat or drink, including water, for a half hour. So the reason for that is because we want to have more of your cells in your spit than water. Because sometimes the test will come back and say, oh, there wasn't enough DNA in the sample. And I'm like, I feel like maybe that patient ate something, in the waiting room before. And they're like, no, it's fine. I've had nothing, or I send them home with a kit and they have something and they're like, oh, I'm sure it will be fine. But yeah, because some people ask me like, oh, is the accuracy any different? I say, no, if we're able to get the DNA, there's no difference in accuracy, but sometimes we just don't have enough DNA to actually run the sample. So that's annoying.
Well, I've heard reports of various operations that require no eating or drinking for some amount of time beforehand. And some patients seem to think that that's just like, I don't know, advisory or something that the doctors do just for the fun of it for some reason, because they're just like, oh no, I'll just have a huge meal before this massive operation. It's like, what are you doing? Like, why?
Yep. Now we got to reschedule that. Everybody's got to wait at the hospital, whatever it is. Yeah.
Just,
definitely.
It's strange. Anyway, okay. So we can get genetic material through a blood sample or through cheek swabs or saliva, and we sort of mix the material, get the cells we need, and extract the DNA from that. So I have a question then about whole exome sequencing, because as you mentioned, basically all the cells in our body have the same DNA, except for the few that don't have any DNA, but genetic material that's expressed is different in different cells. So if you're taking a whole exome sequence, does that not depend on the type of cell that you're looking at? And is that important for any of these applications?
Yeah, that's a good question. So that is a whole nother area of testing. So like epigenetic testing, looking at what genes are turned on and which are turned off. That's going to be very tissue dependent, cell dependent. When we're looking at whole exome sequencing, we're just looking at any genes in the genome, the human genome. So it doesn't matter if, okay, my eyes are going to have different genes turned on than my lips do because they look different, right? They have different pigments. So for whole exome sequencing, we're just looking at any genes that are active. We used to call the other 99% of the genome junk DNA. Like when I was in high school back in 2012, 2013, we would say that was junk DNA. And I always wondered, like, how can it be junk? Like, how can 99% of our DNA be junk? That just seems crazy. And later we found out, all right, we're not going to call it junk DNA anymore because that part of the genome actually does have roles in important roles, but it's just not actively becoming proteins. So it's more like regulatory roles, or it's controlling other genes. So, when we're looking at whole exome sequencing, we're just like, all right, let's look at the active genes. So it's not necessarily active to that tissue, but just in terms of like in our bodies, is it active? Yeah.
Yeah, so that's a topic that I want to do an episode on probably fairly soon, is the so-called junk DNA, or I guess non-coding DNA, I think is the preferred term now.
Yes, exactly.
And as well as epigenetics and control of gene expression, something that is, well, still not very well understood, but there's a lot of work into that recently. This is a little bit of a tangent, but I'm interested in your view on this because when you read about this sort of stuff, you'll see, well, what is it, 1% or so of the DNA actually codes for protein, and then some other percentages are involved in control of gene expression regulation. Some percentage of it's probably structural, like keeping it in the right shape. But there's definitely a lot of other stuff in there, like remnants of old viruses, for example, that have been inserted into the genome, and repeats of short sequences that vary between people. I guess this is just sort of a general question. How much of the sequence kind of matters, and how much of it is sort of this sort of evolutionary leftover, if you like? I mean, I know that that's not a very we don't really know, but I'm just sort of curious as what's your thought on that?
Yeah, I think it's interesting because we look at a technology like CRISPR. I don't know if you're familiar with the genetic editing technology.
Yeah, a little bit.
Yeah. So basically what CRISPR is we discovered this naturally in bacteria, a little over 10 years ago. Well, it was discovered before that, but, a lot of development started happening in 2012 with CRISPR. But so basically what we found was that bacteria have this natural immune system where if they come across a virus, as you said and alluded to, they chop it up and then they keep it in their memory by putting it in their own DNA so that when they come across it again, they're like, oh, hey, we've seen this before. This is an invader. Now we know to get rid of it. So I think that that's interesting when we look at like the evolutionary process, and that even like our mitochondria, one of our organelles, so like little organs in our cells, those used to be not there. That used to just be bacteria. And then at some point, became in our cells very long time ago. So I wonder from like an evolutionary standpoint, as you're bringing up, like, are some of what's in our non-coding DNA, like is part of that, like old viruses that we like fought a long time ago and are keeping track of that. Certainly not an area of expertise for me, but I'm kind of like you. I'm like, that's an interesting area to like look at. And I wonder with CRISPR, if that's going to become more relevant and we're going to learn more about the non-coding DNA and like, what are the other purposes to it? Because there's been a lot of, realization in the last 10 years of like, okay, it's not junk DNA. So what are all the roles to it?
Yeah, I think that it's, well, I guess I don't know exactly what the popular perception might be, but that people know that we've sequenced the human genome. And I think that there's maybe a lack of understanding of how little of it we actually understand as to what it's even for. We're mostly studying a few percentages of it. And there's a lot of mystery about what the rest of it does. And
yeah, we can read it, but we can't understand all of it.
Yeah, exactly.
And I think there's even a small percentage that either we recently finished or we still need to finish. And like part of that non-coding DNA that, when we were done with the Human Genome Project, and it was like a big deal that it was a draft. It wasn't like 100% complete.
Yeah. So I know
I'm still not sure exactly where we are now with it.
I know there are some highly repetitive sections, which are very hard to sequence because you can't figure out it's difficult to figure out how many repeats there are when it's just the same thing over again. I don't know exactly what the status of that is.
Right. I think that's a lot of in like the telomere. So the ends of the chromosomes. Yeah.
Next question I have is sort of related to this, that there's sort of two terms that, well, one might hear in this space: genotyping and gene sequencing. What's the difference between those?
Yeah, that is a great question. And one that I ask a lot of labs when I'm looking at like, which should I order from? Like some of my questions in terms of understanding, like, what are they actually doing in the lab? So genetic sequencing is what we were talking about earlier with, we're going to read through this entire gene and we're going to see, is there any differences in that gene? Now, genotyping is we're just going to look at little hot spots on the gene. So sometimes I think of it like a highway. And if we're doing genetic sequencing, we're driving down the entire highway. Now, with genotyping, we're only going to pop in on the exits. So we're not going to actually drive through. We're just popping in on the exits, and like looking at hot spots. So a lot of genetic testing, when we were starting with this and years ago, a lot of it was genotyping because we'd say, well, we know the most common mutations, which we also call pathogenic variants. That's more of the scientific term now for a mutation. So we used to say, all right, well, this is the most common mutations. Let's just look for that. Let's not take the time to look through the entire gene. And there's advantages to that. Obviously, it's going to be cheaper if you're just looking at certain spots on the gene. But there's a lot of disadvantages because what if there's a mutation elsewhere on the gene and you're going to miss that because you're not even going to look for it? So I think that's something that's changing a lot. I still see some providers ordering genotyping, but that's not something that I do anymore because to me, the technology is beyond that. Let's sequence the entire gene for the ones that we're ordering.
So just a point of clarification here, you're talking about sequencing an entire gene. So is the way it works, you order a set of specific genes that are then sequenced. You don't sequence the whole genome or even the whole exome. You're just looking at a set number of genes, but you sequence that entire gene. Is that how it works?
Right. So it depends what someone orders. So
Yeah.
if we were ordering a whole exome, then we're just going to be sequencing all of the active genes as we talked about. Whole genome, everything. We're going to sequence everything. And then, I haven't ordered any of those. So only as a student, certainly looking at doctors and other genetic counselors doing that. But for me, I've ordered genetic panels. So that's selecting specific genes that I'm going to have my patient be sequenced for those certain genes. And that's because I work in prenatal. So when I'm doing carrier screening, I'm looking at the parents to see, okay, are they a carrier of a condition like cystic fibrosis, sickle cell? I want to sequence those genes. I don't care about the rest because I'm in a prenatal setting. That could change in the future. But yeah, it definitely depends on like why you're ordering the test, what you're looking for. Did you know PerkinElmer Genomics was one of the first laboratories to offer whole genome sequencing on a clinical basis? Whole genome sequencing can maximize clinical diagnostic yield for patients. With a turnaround time of 6 weeks, PerkinElmer's whole genome sequencing test is designed to provide access to additional valuable information compared to an exome. PerkinElmer Genomics provides one of the world's most comprehensive programs for detecting clinically significant genomic changes. PerkinElmer Genomics delivers knowledge that can empower health. PerkinElmer is a global leader committed to innovating for a healthier world. Join their mission at PerkinElmerGenomics.com. Also, listen back to episode 176 with PerkinElmer's Dr. Madhuri Hegde, where we explored the power of whole genome sequencing. Discover all that PerkinElmer Genomics has to offer at PerkinElmerGenomics.com. Do you know the power of polygenic risk score? Polygenic risk score, or PRS, enables healthcare providers to more effectively help patients lower their risk of life-threatening diseases with the latest breakthrough in clinical genomics. Allelica has launched the first PRS test on the market to calculate Ancestry-specific PRS for breast cancer, prostate cancer, coronary artery disease, type 2 diabetes, and Alzheimer's disease, and delivers the results in a clinical-grade report. The PRS test is a laboratory-developed test that is performed in a CLIA-certified, CAP-accredited genetics lab. Here's just one example of why this test is so important. If a patient with breast cancer tests negative for BRCA1, BRCA2 genetic mutations, Allelica's PRS test may be able to identify other key genetic mutations which played a role in the cancer development. Learn more about PRS in our previous interview with Allelica CEO and co-founder Giordano Botta on episode 168 of DNA Today. You can order the PRS test at order.allelica.com. For 25% off and free shipping, use code DNA Today at checkout. Allelica, empowering the next generation of clinical genomics.
Yeah, so just for some context here, well, you might know the number better than I do. So the current estimate for the number of genes that humans have, the number I have in my head is 30,000. Is that about? I
think it's around like 20, 30,000. Yeah, that sounds right.
I've seen some different estimates. I recall that before the Human Genome Project, scientists had estimated it was much higher, like 100,000, but then it was discovered that there were actually far fewer than they'd expected, which itself is interesting. How we really
thought highly of ourselves. We're like, we must have so many genes, because they were comparing it off of other genomes too. And they were like, well, if this animal has this many genes, we're way more complex. We must have way more. And then we were like, oh, we don't. So it was a bit of like a humbling moment, I think, for the human race.
Yes, partly. Although I guess, again, this is a little speculative. My kind of take on that is that probably control of gene expression, genetic regulation is doing a lot more of the work than we thought. I guess naively, it's like, oh, to do more things, you need more genes, but maybe it's actually just that you have more complex and intricate control of the regulation expression.
I think you're on the right track there, because otherwise we would have that correlation between like, all right, if a animal is more complex, then they must have more genes. But it's like we even see with like fruit, like certain fruit have like crazy amount of chromosomes, and we're like, all right, so there's something that we're not quite understanding there. And it probably lies in epigenetics there.
Yeah. Anyway, so just coming back to what we were talking about before. So if there's about 3 billion nucleotides, then, and if you do a whole genome sequence, then obviously that's a lot. A whole exome sequence, that's about 1% of that. So that would be, 30,000, I think. 30 million. And then, it's easy to get the orders of magnitudes wrong. And then if we've got about 30,000 genes, so you pick a few of those and sequence those. a gene is, I don't know, 1,000, a few hundred long, depending on, obviously it varies by the gene. So that's just giving a bit of a framework for people to think about the numbers we're talking about here. So obviously it varies depending on exactly what you're ordering the test for and what the purpose of that is. And actually that makes a good move to the next question, which is applications of genetic testing. So far we've sort of been talking a bit about the science of it and how it's done. We've mentioned sort of vaguely that there's various health applications, but let's look at that a bit more specifically. So what are some of the reasons why medical practitioners would want to order genetic testing? And also there's the new field of direct-to-consumer genetic testing, which I want to talk a bit about as well. So what are some of the applications there in either of those?
Yeah. So there's so many areas of healthcare where we're ordering genetic testing. I can speak to my area first and then kind of fan out from there. So as a prenatal genetic counselor, I kind of mentioned before a panel of genes. So if I was doing carrier screening for someone that's pregnant and their partner, for that, I'd be looking to see, are they a carrier of the same condition? So these are autosomal recessive conditions for the most part. And I'm looking to see for those conditions and for most, we have 2 copies of each gene. So if one copy doesn't work, then that person is a carrier of that condition. They have a backup copy. So they're, for the most part, healthy. They probably don't have symptoms from it. If both a patient and their partner or the sperm donor, egg donor, whatever the situation is, if they are also a carrier for that same condition, they have a chance for passing that down to a baby, where a baby could inherit both of those non-working copies that have a mutation, and then baby has no working copies and has the condition. So with carrier screening, I'm looking to see, are they a carrier for anything? And are the couple matching for a condition? So that's a case where nobody has a disease, nobody has a condition, but we're, more doing on the preventative side. So again, that's like more of the gene panel.
Sorry,
just go ahead, James. Yeah.
Just to clarify with that. So the key, the way I think about it is the key issue there is that parents can be a carrier for a condition that they don't themselves exhibit. So that's the difference that we talk about between genotype, which is their genes, and the phenotype, which is the characteristics that they express. And so they may have no idea that they're carriers for a particular condition, but potentially if, say, they're both carriers and it's a recessive condition, then if they both pass that copy of the gene on to their child, then the child could exhibit the trait. So that's the sort of thing that we can test for genetically that there's not really any other way to check for.
Right. And as you said, most people don't realize they're a carrier for a condition because most times carriers don't have any symptoms. So some people might say like, oh, well, I'm healthy. Like, I don't see the point of doing carrier screening. They say, well, most people, if we look at enough conditions, most people are a carrier for something, because we're testing for hundreds of conditions. If we're only testing for a couple, all right, you're probably not a carrier for those. But the more conditions we're looking at, the more likely you're a carrier for something. Usually it's different from your partner, but sometimes it's not, which is why we do the testing.
Yeah. And sorry, go ahead. You were then going to talk about other applications.
Yeah. So another application that I order is for people that are pregnant. There's a test called non-invasive prenatal screening. And this is really cool because we found out that coming off the placenta are cells that float in the pregnant person's bloodstream. And those cells tend to pop open and release the DNA. So by taking a blood sample, we can isolate that DNA and then screen for genetic conditions. And these are random conditions, not inherited like I had been talking about with carrier screening. So these are random conditions, most commonly Down syndrome, where we have an extra chromosome 21. So it's a really interesting technology that, we discovered like, wow, there's actually cells from the placenta in the pregnant person. What if we can use that to screen for conditions? So that's a test that has become much more popular. It was clinically available about 10 years ago, but the last few years has become much more popular.
Yeah. So for these, so we've talked about genetically inherited traits, which you can check the mother and the father to see whether that was likely to be an issue. But unfortunately, that it doesn't stop there, because there's also the possibility of genetic mutation where basically random events can happen, which can then lead to characteristics or disease states in the offspring. And so what you're saying is that there are actually there's genetic material which enters the bloodstream of the mother during pregnancy, which we can then test for and see if there are any of these conditions in the offspring. One question I have is if there are, I think this is actually a broader issue, which I confess I don't fully understand. How does that work with respect to the mother's immune system? If there's foreign material, wouldn't that be attacked by the mother's immune response, or is that suppressed during pregnancy? I'm not quite sure how all that works.
Yeah, that's a good question. So during pregnancy, the body does have a lower immune response because otherwise we'd probably fight the pregnancy because our body would be like, what are all these cells growing? Like thinking like, is it cancer? Right. So in general, your immune system is lower during pregnancy. But yeah, I'm also a little curious because I don't fully understand like how we can have cells in us that are not ours. I mean, 50% of it is our genetics if it's our biological child, and our genetic child. But what's interesting is that the cell-free DNA, so when a cell pops open and releases the DNA, that is after a person gives birth, that clears from their system very quickly. I've heard like hours, like that quickly. But the cells that are still intact, that can stay in a person's body for years.
Oh, wow.
Yeah. So I remember I was reading a book by Carl Zimmer, and she has her mother's laugh. And I ended up interviewing him on my show. And I just remember being like amazed. It was talking about some case where they were able to find in people that they had for people that are 46XX. So like a traditional female karyotype, that they were able to find some Y cells, some cells that had the Y chromosome. And they're like, well, what is this? And they're finding it was the origin was those people's sons from previous pregnancies, that their cells were still there. And I was like, that still amazes me to this day. And I read this book like 4 years ago.
That's crazy. I've never heard of that before.
Yeah. Isn't that wild? But the technology takes advantage of the cell-free DNA. That's how it works. So if we were looking at cells that are intact, it would fall through. We wouldn't be able to test it.
Yeah. Oh, that's really interesting. Okay. So far we've got testing for recessive conditions in parents and also testing residual DNA from pregnancy. What other applications are there for genetic screening?
Yeah. So going outside of my practice, in the cancer realm, we can look at genes that are known to protect us from cancer. If there's a pathogenic variant or mutation in one of those genes, it lowers our protection for cancer. So then in turn, it increases our risk to develop cancer. So if our protection for cancer is lower, we're naturally going to be more likely to develop cancer. So there are certain genes that we can look for. The most common is BRCA1 and 2. So these are genes that people may know because of Angelina Jolie a few years ago was in the news because she shared that she has a pathogenic variant in a BRCA gene and ended up having preventative surgery so that she was reducing her risk to develop cancer. So I think that's one that some people might have heard of before. And so, yeah, cancer is an interesting area because that's another area where you can either be doing it preventatively. So say your mom had breast cancer at 35. You're like, okay, well, that to me, that's high risk. Let me see if I have genetic change in me that also increases my risk for breast cancer. And then you can also be doing it after you're diagnosed with cancer so that we understand, okay, what led to the cancer to happen. So that's a test where we can do it both ways.
Yeah, right. I want to talk a little bit more about that. Before maybe moving on, there are a couple of other issues that I just wanted to mention. So listeners are probably also familiar with other uses of genetic testing. I guess it still counts as genetic testing. It's not diagnostic testing though. So this would include for forensic use, like in terms of for solving criminal cases, as well as for genealogical tests, which are increasingly popular for people to find out about their ancestry, and for paternity testing, which is obviously one people will be familiar with. And for those usages, at least for forensic and paternity, my understanding is that we only need a relatively small number of markers, which vary between persons, and that doesn't need. I think those aren't even genetic markers in the sense that they're from genes. I think that they're just from non-coding DNA. But just sufficient to basically identify persons. So it's rather different from the diagnostic testing. I know that that's not what you work on. I thought I would just mention that because listeners may also be thinking about that. But
yeah, it's a cool area.
Yeah, all of these applications have increased dramatically in recent years because of the development of technology. So now I wanted to ask you a question about direct-to-consumer genetic testing, which has expanded very rapidly in recent years. So this is when consumers directly order various genetic tests from various private companies, not like directly or necessarily through a medical practitioner. So certain companies have come under criticism for making claims about the medical benefits or applications of these sorts of tests. And it's a tricky area, partly because in many cases, it's sort of not known whether or not there's any medically actionable benefit from knowing whether you're predisposed to a particular condition or not. Because often we're talking about probabilities in these cases. It having this variant slightly increases your risk of this type of cancer and so forth. And that sort of information is sort of quite abstract and often difficult for people to, well, it's often difficult enough for medical practitioners to understand, let alone people who don't have that sort of training. So I'm just interested, what are your sort of thoughts about this sort of direct-to-consumer, health-focused genetic testing and some of the issues surrounding that?
It's difficult because a lot of people don't fully understand what the testing can tell you and what it can't tell you. I think if people understood that, I'd have a lot less issues with the direct consumer testing. And there's even a testing that is like in between medical grade and direct consumer where people can order it themselves, but there's a healthcare provider directly involved with them. Right. So it's kind of like a middle ground too. But for these direct consumer where you buy the kit, it either gets shipped to your house, you're picking it up at Target, Walmart, wherever, and you're just sending it off and then you get results in your email or through a portal and there's no person telling you results. So all those direct consumer, I think it's important to know that it's not going to be as extensive as testing you're going to get with a healthcare provider. So using the breast cancer genes that I was talking about before, BRCA1 and 2, I think that's a good example because, and I'll be frank, 23andMe does will test for these genes. And a lot of people know this company. It's one of the biggest direct consumer companies.
Yeah, they've received a lot of press coverage in the last few years.
Yeah, definitely. And so with the BRCA genes, they do genotyping like we talked about. So they look at the 3 most common mutations in BRCA genes. Now, these are mutations that are common in the Ashkenazi Jewish population. And the problem I have with it is if a person does the testing and it comes back negative, like, okay, didn't find any mutations, they may not understand that it's just that they don't have those 3 mutations. It didn't look at the rest. They could have a different mutation in that BRCA gene and think now, oh, I'm negative. So I think if people understood that and said, okay, well, I'm only negative for these 3 mutations, maybe now I want to go and see a healthcare provider to see, do I have a mutation elsewhere in these genes or a different gene related to breast, ovarian cancer, prostate cancer, all of that. So I think there's a time and a place for it. I think the Ancestry is very interesting as well, but it's also something that there's a big discrepancy between people of European descent and people of non-European descent. We have a lot more in databases for people of European descent because that's how genetics started. We started with a lot of European genomes. So we understand those variants better. We understand the spelling of those genes better. But if someone is of non-European descent, the ancestry results they'll get back is going to be much more general. So I'm of European ancestry and one of, branches of my family is from Ireland. My testing gets so specific to say I'm from East Cork, Ireland.
Oh, wow.
So it's very specific, right? And lucky for me, that lines up with family stories. I don't have to tell anyone, oh, we're actually not from there, which is a whole nother thing, right? But, people have more recent African ancestry. It's probably going to give them more of a general, you're from this area and not necessarily pinpointing, to a certain county or something. So, and this is an area that is, we have huge diversity issues in all genetic testing because of that. Ancestry people are more familiar with, but this is a problem where we have these genetic changes and we don't understand, okay, is this making the gene not work or is it human diversity because our research pools are lacking diversity?
Yeah, and it's quite difficult because there are many genes and many variations on those genes and many possible conditions. And also, as we sort of mentioned, for a lot of, especially things like cancer and heart disease and so forth, it's not like one, and I think people don't understand this very well. It's generally not the case that a particular variant is definitely going to give you a particular disease.
Yes.
There are some diseases like that, but for the most part, it doesn't work that way. And it's an issue of, well, it increases the risk or possibly decreases risk by a certain amount. And the thing is, that it gets even more complicated than that because it could be an environmental interaction. So it may be, for instance, that if you're, I'm just making this example up, but if you're a smoker, it increases your risk of certain types of cancers, but not if you're not a smoker. Or if you have a poor diet, it increases the risk, but for people with a good diet, maybe it doesn't. And so this sort of information is very difficult, as I said, even for medical practitioners to understand, let alone sort of layperson. So I guess I have a concern about how actionable some of this medical information is. So if I found out that I was at a bit of a higher risk to develop certain types of cancer or heart disease, it's sort of not clear to me what I would do other than, eat healthy, exercise, and the usual advice anyway. So I mean, I gather that probably for some conditions there is actionable medical advice. I know that there's a list of about 60 variants that I forget the organization, but in the US, they've recommended that these be reported.
ACMG. Yeah,
Yes.
ACMG in the US at least. I'm less familiar with Australia, to be honest.
Yeah, I don't know that the case here is probably similar, but that they,
right.
Well, just to give a bit of background there. So there's a further complication here in terms of, we've been talking about whether someone seeks out this genetic information, but there's a further issue that if a diagnostic test has been conducted for some other reason and they sequence a bunch of genes or the whole genome or whatever, and they find other variations, should they tell the patient? The issue is there, well, if it's important, you should tell them, but how do you quantify important? Like, is it medically actionable? What's the risk and things like that? And we often don't have very good data. So there's this list of around 60 particular variations that, as I understand, is recommended. I don't know that they're legal or they have to, but I think it's a recommendation that they be reported. I don't know a lot about, though, what medical actions sort of can be taken on those. So what's your knowledge on that, or just general thoughts?
Yeah. So usually that's coming up, and it's if I'm remembering right, ACMG 59 genes. Yep. It could have been updated since last time I looked at it, because I haven't been in that area for a bit. But so if someone's doing that whole exome sequencing that we talked about, or whole genome, and they find a pathogenic variant mutation in one of these 59 or so genes, then hopefully ahead of time, the patient has sat with a healthcare provider and decided if something comes up in one of those genes, they either want to know about it or don't want to know about it. So hopefully they had informed consent before they even got results. If they decided they wanted to know about it, some of these genes might be, as we talked about, like a BRCA gene mutation, where, okay, they have an increased risk for breast cancer or ovarian cancer, prostate cancer. So it's different cancer risk there. And so in those situations, people can have the option of doing more screening. So for breast cancer, they might be doing breast MRIs, ultrasounds, possibly mammograms, definitely, so that they're screened more regularly than someone that has an average risk for breast cancer. There's also options to have, as I mentioned with Angelina Jolie, of having preventative surgeries. So to remove breast tissue before you even possibly develop cancer, just to reduce your risk. And you might never have developed breast cancer in your life, but some people look at it, well, I have an 85% chance. All right, that's pretty high. Some people decide to do the preventative surgery. So that's called a prophylactic bilateral mastectomy. So a bit of a mouthful.
Have such long names.
Very long names. It took me years, I have to say, to get all the lingo down and pronounce it right and everything. But so there's certainly, for some conditions and some genes that we find in mutation, there are actions we can do to either reduce our risk or to know, like, you have this diagnosis, you may want to plan something in your life of if you know that you're at higher risk of developing a condition.
Yeah, I guess each patient is going to be different there. I think that the difficulty is in getting informed consent, because it's hard to give the relevant information to people to understand all of these sorts of risks. For me personally, and again, this is just sort of my view, I probably wouldn't want to know about any condition unless it was something that I could do something about. I mean, I suppose I agree. There's always the question, like, well, if you knew you were going to die on this particular day, would you want to know that? That's an issue. But more generally, I feel like, and probably this is going to be true for a lot of people, that there's the question about increased worry and stress if they think that they have a risk for something, especially if the increased risk is maybe marginal compared to lifestyle choices, which I think is a factor people, I think, overestimate the effect of genetics. I don't actually know if there's data on this, but
I feel like it's true,
choices. And I think that that's something that the medical field is sort of trying to be more cognizant of, is doing more about sort of encouraging positive lifestyle changes instead of just focusing on the genetics. Obviously, though, that plays a role, and they can interact, as we mentioned before. But yeah, but there are some cases where action can be taken, and in that case, then this is useful. I think that there's also, people have a perception, many patients, I would say, have a perception that information is always good and that it's good to know, it's good to screen and things like that. But increasingly, I think the medical community is coming to realize that often that's actually not the case. For example, if conditions have a very high, if a condition has a high, a relatively high baseline, relative to the sensitivity and specificity of the test, then you can get a large number of false negatives. And then you can end up spending large amounts of money and time and stress getting tests and biopsies and scans and whatever for conditions that you don't actually have or for which the risk is relatively small. And all of these things have costs associated with them. So there's a balancing act there about how medically useful the information is and what the risk is balanced against the costs and the stress. And I guess I would add as well, just sort of attention taken away from things that might be more helpful, like lifestyle improvements.
Right. Yes. Yeah, definitely. And I think another aspect of that is like looking at informed consent, like in our healthcare systems, we don't necessarily have time to sit with a patient. I mean, back when I was a student and I was observing genetic counselors consenting patients into doing this whole genome, whole exome. I mean, it would take a long time to say, okay, do you want to know this? Do you want to know that? Like, This is how the testing works. This is what could come up. Are you sure you want to do this? And I think it is really important, like, to have informed consent, because when I sit down and talk with patients about, as we talked about in the prenatal setting, carrier screening and the non-invasive prenatal screening, I talk it through and I say, this is what we could find out. Patients will say, all right, well, what do I do with this information? We talk that through. And sometimes at the end of our conversation, patients say, well, thanks for going through all that. I'm glad we did, because I don't want this testing. And I'm like, I'm really about it too, because other places might just draw their blood and say, we'll let you know. And they don't even know what they're getting. Yeah. So I think it's so important that we take the time, we make sure a patient understands what the testing is and that they actually want to do it. Because the last thing I want to do is make a decision for a patient. That's not my job. My job is to educate the patient and help them make a decision that's best for them and their family.
Yeah, one of the things that we've talked about on the Science of Everything before is different cognitive and psychological biases. And I think that it's interesting how those potentially interact in the health space about people's estimates of risk and the value of information and things like that. So I think going forward, we're going to need a lot better research that sort of looks at things from an interdisciplinary point of view about how people respond to information and how they, what sort of medical information they seek out and what's sort of motivating to people and actually is sort of helpful for them and what isn't. So I think that there's, I guess broadly, the genetic age is only, well, I don't know, if you date it from the beginning of from the sequencing of the human genome, like 20-ish years old, we're still sort of learning how to do an information-rich genetics age. And I think that going forward, we'll hopefully develop better systems and get sort of used to having this information. But until then, I think we're groping around a bit and trying to work out how to do it well.
Yeah, I think so. Polygenic risk scores, or PRS, are a powerful way to identify an individual's risk of common disease by analyzing up to millions of genetic variants spread across the genome. Allelica has launched the first PRS test on the market to calculate ancestry-specific PRS for breast cancer, prostate cancer, coronary artery disease, type 2 diabetes, and Alzheimer's disease, and delivers the results in a clinical-grade report. Order your test today at order.allelica.com. For 25% off and free shipping, use code DNA today at checkout. Again, that's order.allelica.com. Allelica, empowering the next generation of clinical genomics. PerkinElmer Genomics is a global leader in genetic testing, focusing on rare diseases, inherited disorders, newborn screening, and hereditary cancer. Testing services support the full continuum of care from preconception and prenatal to neonatal, pediatric, and adult. Testing options include sequencing for targeted genes, multiple genes, the whole exome or genome, and copy number variations. Using a simple saliva or blood sample, PerkinElmer Genomics answers complex genetic questions that can proactively inform patient care and end the diagnostic odyssey for families. Learn more at PerkinElmerGenomics.com. If you enjoyed DNA Today, especially episode 139 with Dani Shapiro, who discovered she was donor conceived, then you'll also enjoy more shocking discoveries from DNA testing and a new podcast I love, Biohacked Family Secrets. It's a brand new podcast all about hidden ancestry, medicine, and technology. The show follows the children of anonymous sperm and egg donors, some with dozens or even hundreds of half-siblings, who accidentally learn the truth because of commercial DNA tests like 23andMe. The podcast drops some bombshells and shows how new technology is unraveling the deep, dark secrets of the baby business. Along with scandalous personal stories, Biohacked Family Secrets also reveals unexpected ethical, scientific, and political questions. It's a vital listen for anyone who loves wild stories about science, tech, and history, or even just messy family drama. You can find Biohacked Family Secrets wherever you're listening right now. New episodes drop every Wednesday.
So before we finish up, I guess that actually leads well into the last question. What do you think about the future of genetic testing techniques and genetic screening, and also future applications as technology changes and develops? It's obviously changed a lot even over the last 10 years. So what do you think is going to happen in the future?
Right. I think in terms of how we're changing with genetic testing, I do think that a lot more people are going to get either whole exome sequencing or whole genome sequencing, because a lot of the testing is, okay, we'll do a gene panel. We'll look at these certain genes, but over time, isn't it going to be more cost effective, as we were mentioning earlier, to just look at everything and then keep referring back to that over that person's life? So, if someone's young, say, all right, let's sequence their genome, let's sequence their exome, and then we can keep referring back to that instead of throughout their life, keep ordering different genetic tests. I think we're going to get to a point where genetic testing has that cost effectiveness to be able to say, all right, it actually is cheap enough to do this now. And it's going to start by people deciding to spend the money to do it. And then eventually, hopefully, we'll be a lot more patients will have access to it. But just like anything in life, as technology develops, people that have money are going to be able to do this. And then slowly it will trickle down. The more people that order it, that cost and demand will keep lowering the price for genetic sequencing. And so I think that's where we're headed in the future. But it's going to end up bringing up more questions, because the more people that have sequencing, we're going to say, well, what about all these genetic changes? So we need a lot more research in terms of figuring out, as we said, just because we have sequenced the human genome doesn't mean we understand all of it. We're really at the beginning of the understanding. And I think that's why it's such an exciting field, and one to keep your eye on in genetics, because there's just so much that develops so quickly. So hopefully we see that, as well as affecting people and just being able to get more genetic testing and to have that information to help them with decision-making in healthcare.
Yeah, I'm looking forward to seeing how this develops as well. I guess one possible outcome, which to me seems almost inevitable in the long term, although maybe that's too strong, and I'd be interested to hear your thoughts, is that basically, at least in developed countries, whenever a child is born, the whole genome is just sequenced, and that information is stored somewhere, hopefully securely, and then can be used as a reference point for all sorts of medical conditions later in life. Obviously, we're not at that point yet, but if it becomes cheap enough and accepted enough, I could see that happening. I mean, do you think that that's likely? Or would that be a good thing? Is there too many privacy issues? Like, what do you think?
Yeah, I think it's interesting. There's something called newborn screening, where in the US, all babies have newborn screening unless a parent opts out. And that's quite a process. I've never heard of a parent opting out. So where through a heel prick test with the baby, they're tested for certain conditions that, if we diagnose very early, can change a baby's life, possibly save their life. So obviously this is important. So I've asked a lot of guests myself, like, do you see whole exome or whole genome sequencing replacing newborn screening? Like, what if we just learn this when someone is just born? And as you said, have that follow them. And I think there's a couple hurdles we have to get through with that. Some of which are, what about these adult onset conditions? Like we both said, we don't really want to know about a condition that we can't do anything about, right? So that's our own personal choices. We can't make a decision for a baby. What if they don't want to know any of this? So I think we'd have to come up with a way to be able to unlock information as that child gets older. So at first, only know conditions that are going to affect them in childhood, teenagehood. And then once they turn 18, then they could learn about adult onset conditions. Yeah. But the problem is, like, all right, who's going to hold this data? How are we going to make it secure? Who's going to let them know about these new conditions in 18 years? Those people that would call you with results are not going to be in that same job 18 years later and have that set up. So I think in this ideal world, theoretically, it's really great and interesting, but there's a lot of issues we'd have to work out. But I think we'll get there at some point. It may not be for our generation, but maybe a generation below us or the next one or something, where we have this as like a standard in developed countries.
Yeah, one thing I just listened to an audiobook recently about the Bay Area rapist, I think it's called. So it's the
Golden State Killer.
Oh, sorry. Yeah. I think they're the same, but yeah. Anyway, so I listened to an audiobook about this, which was written by a woman who was basically just investigated it as a hobby. And she actually died tragically just a couple of years before he was actually caught using genetic technology. Oh, wow. And what was interesting to me is that this might sound like a tangent, but linking this back in, the original crimes were committed during the 70s and 80s where gene technology just didn't really exist. But material was collected, which then later was subject to genotyping, which allows you to basically, if you have another sample from that person, match. So you can tell with high probability who it is. But you need the point is you need a sample from that person. Now, they actually ended up catching him because they found a match in, I think it was an ancestral DNA database from someone who was his distant relative, and they were able to
a distant cousin or something that. Yeah.
And they were able to then build different family trees based on that, and then using other records, and gradually they were able to, by process of elimination, to sort of reduce it to just him. And I think that's just amazing that was possible, that decades after the fact that you can use genetic material to find someone.
Right.
And again, I guess that this is sort of the criminal investigator fantasy. It's like, if we just had everyone's markers on file, like for the whole population, you could just solve so many crimes basically instantly, as long as there's any genetic material. Now, of course, then you have, well, what about the privacy concerns there? And part of me thinks, well, look, if you just store the markers, there's very little you can actually do with that information other than use it to identify a person. And I know that, maybe that's too quick, but I guess the point is that, not to give a definitive answer on this by any means, but I think that in that case, in the criminal justice case, similar to what we were talking about in the medical case, we haven't as a society fully realized or worked out how to deal with having this information and all of the potential upsides as well as downsides that it represents. And I'm interested to see where that's sort of going to go in the future. So maybe if you want to close out on a few thoughts on that.
Yeah, I think it's interesting because that brings up a lot of privacy of when I have used direct consumer tests, I spit, it's my spit. It's my DNA. So I'm consenting to send that off for the company to sequence that or, genotype, I guess. And then now my DNA is in the database and it's labeled as Dineen. I didn't use a pseudonym, like maybe I should have. And it's funny because I brought this up at Christmas Eve with a bunch of my cousins. Right. And one of my cousins goes, well, you didn't have consent to do that. That's my DNA too. Right. Cause we're biological cousins, we're related. And, and he's a sarcastic, funny guy, but here's the point, right? So when I send my DNA, if my, funny, great cousin, if he goes out and, commits a crime, what if they could use my DNA to help figure out, oh, it was Kira's cousin, right?
Yeah. Well, they can. They've done it before.
And so, Golden State Killer case right there. So I think that does bring up that it's with all of this data, we don't know exactly how it will be used. And when we send it off to a company, that person probably doesn't fully understand that company's privacy policy. The company I sent it to could end up selling that DNA to another company. And now they have that information. So I think that's something where with genetics overall, we're okay, where could this information go? And how are we going to use it in the future? It develops so quickly, even looking at things like we used to say, oh, it's an anonymous sperm donor. That there's no such thing as anonymity in genetics anymore. Those people that were told, oh, it's anonymous, now are getting identified through these databases of oh, I'm your biological son, I'm your biological daughter, or whatever. And they were I never signed up for this. It was an anonymous donation. So I think that's something to keep in mind with all of this is we don't fully understand where we're going with genetics and everything that we can uncover from it, which I think is mostly exciting, but also a little bit scary.
Yeah, well, I agree. And I think it's one of a good reason to be educated about some of the science behind it so we can make more informed decisions and voting decisions and health decisions and also just giving advice to other people. So it's been a pleasure. I've learned a lot and hopefully listeners have too. So thanks, Kira, for joining us today.
Great. Thank you for having me, James. And for anyone that wants to check out my podcast, it's DNA Today. So you can search that on any podcast player. It will pop up. For more information about today's episode, visit dnapodcast.com, where you can also stream all episodes of the show. We encourage your questions, comments, guest pitches, and ideas. Send them all into info@dnapodcast.com. Search DNA Today on Twitter, Instagram, YouTube, Facebook, so you can connect with us there. And a favor, please rate and review the podcast on Apple, Spotify, or wherever you listen. This truly helps us climb the charts and allow more genetic nerds like yourself to find the show. DNA Today is hosted and produced by myself, Kira Dineen. Our social media lead is Corinne Merlino. Our video lead is Amanda Andrioli. Thanks for listening, and join us next time to discover new advances in the world of genetics.
The genes of you and me, the genes of you and me, they're all made of DNA. We're all made of the same chemical DNA.